Welcome everybody. I think we'll get started. Thank you all for coming along to this 5th Lloyd Cox Memorial Lecture on this cold and wintry night. Tonight's event is jointly hosted by the Robinson Research Institute here at the University of Adelaide and the Lloyd Cox Research Foundation. I'd like to begin by acknowledging the land that we meet on today is the traditional land for the Kaurna people, and that we respect their spiritual relationship with the country. We also acknowledge the Kaurna people as the traditional custodians of the Adelaide region, and that their cultural and heritage beliefs are still as important to the living Kaurna people today. My name is Professor Sarah Robertson, and I'm the Director of the Robinson Research Institute. And I'd like to welcome everybody and thank you so much for coming along tonight. And just start with a quick couple of housekeeping items. If there's an emergency, please walk up the stairs, orderly fashion please, no pushing to the front. Exit the building, walk along North Terrace towards Samri, which is in that direction. And if you'd like to use the bathroom, there is one here on the ground floor in our fabulous AHMS building. It's up the far end there next to the Bean Bar Cafe. So we're absolutely thrilled to be hosting Professor David Haig from Harvard tonight. And next, I'd like to introduce our Vice-Chancellor, Professor Peter Rathjen, who's going to tell us a little bit more about David and introduce the night's events. Peter, thank you.
Thanks, Sarah. And I think it's my official job, actually, to welcome you here tonight to the 5th Lloyd Cox Memorial Lecture. As Sarah said, it's jointly hosted by the Robinson Research Institute and the Lloyd Cox Foundation. We are extremely proud at the University of Adelaide to be associated with both of those bodies. I am particularly pleased tonight to welcome Lloyd Cox's family. His son and grandson, David Cox and Sam, his daughter, Di Cox, and her partner, Max. Thank you for coming. Universities build across generations. It's wonderful to have the legacy of Lloyd Cox with us on this important occasion. The Robinson Research Institute is one of the University of Adelaide's premier research institutes. It's got more than 350 members working across human reproduction, pregnancy and child health, all focused on giving children the best possible start to life. Last year was the Robinson Research Institute's 10th anniversary. For more than 10 years, we've watched as Robinson Research Institute members work together with the public, with industry, and with the health sector to deliver world-class advances in human reproduction, pregnancy, and child health, improving the health and well-being of children and families over their life course and across generations. The research excellence that we see here today builds on decades of leading research that started with the appointment in 1958 of Lloyd Cox as the university's first professor of obstetrics and gynecology based at the Queen Elizabeth Hospital. Lloyd Cox had previously worked in New Zealand and the UK, and his vision was for the department to be a world leader. He had a strongly held belief that science and medicine should collaborate closely, and that established the foundations for future success. This culture of collaboration has successfully continued under successive leaders. Lloyd Cox wanted obstetrics and gynecology to be a science-based department, and he was the first person to bring infertility as a medical issue to the attention of mainstream medicine. He encouraged people to look at new ideas and to develop new techniques. During his time as head of O&G, Lloyd Cox appointed Ron Cox as a biochemist. This was the first department of its kind to appoint a scientist. It also established the first frozen semen bank in Australia and the first sperm donor program. It's been a pioneer for decades. Professor Cox retired in 1984 and was replaced by Professor Geoffrey Robinson, who we are delighted to welcome here tonight. He was head of department, and Professor Colin Matthews, also here tonight, was appointed as Professor of Reproductive Medicine. The great work continued. The great tradition of great men and great leaders stayed with us. So tonight is a memorial to Lloyd Cox. And in a fitting way, this lecture series brings some of the most inspiring researchers from around the world to Adelaide to present on thought-provoking topics around early life health. And that brings us to our speaker tonight, Professor David Haig, evolutionary biologist and geneticist, the George Putnam Professor of Organism and Evolutionary Biology at Harvard University. He has a particular interest in genetic conflicts within individual organisms, as exemplified by genomic imprinting. His work has covered everything from maternal-fetal conflict in human pregnancy to the evolution of plant life cycles. His major contribution to the field of evolutionary theory is the kinship theory of genomic imprinting. Professor Haig comes from Australia. He undertook his Bachelor of Sciences and his Honours and PhD in Biology at Macquarie University in Sydney, then moved to the UK for postdoctoral work at the University of Oxford. He was then nominated for the Harvard Society of Fellows, and he's worked at Harvard University ever since. We're privileged to hear from him today. Please join with me in welcoming Professor David Haig.
So hopefully the sound is on. After that introduction, I'm feeling a little bit nervous, but that's okay. It's wonderful to be back in Adelaide, and I thank the Cox family for making that possible. I first came to Adelaide as a teenager back in 1973 or something like that. So that's telling you a little bit about my age. In fact, I'm 61. I'm not a particularly healthy individual, but in all those 61 years, my heart has never missed a beat. My kidneys, liver have functioned admirably well. And this is something we know about physiological systems, that they are in extraordinary feats of adaptation, efficiency, second by second, minute by minute, year after year. But somehow, pregnancy is different from other physiological systems. That in the brief 9 months of pregnancy, there's a high frequency of all sorts of physiological problems. And why should that be the case? Because producing a successful child is central to reproductive fitness. Shouldn't pregnancy be more efficient than other physiological processes? And so my talk today is going to be about why pregnancy is different. I think I'll start here. This is expressing the problem that we know about. So this is from John Whitridge Williams' textbook of obstetrics, the major textbook of obstetrics in North America, first edition. And he wrote, from a biological point of view, pregnancy and labour should be considered normal physiological processes. But it is apparent that the borderline between health and disease is less distinctly marked during gestation than at other times, and derangements, so slight as to be of but little consequence under ordinary circumstances, may readily give rise to pathological conditions. We all know that when a woman becomes pregnant, she starts having doctor's appointments in anticipation that something might go wrong. So here, I like this picture. This is a Nepali woman, and there are a lot of demands on her body. She's hard at work, she's carrying a fetus, and she's also carrying a toddler. And her time, energy is divided between all of these tasks. And pregnancy is different because all of these individuals sitting in this, genetic individuals sitting in this picture, have different fitnesses. What is best for the fitness of the mother is not necessarily best for the survival and reproduction of the fetus. And there's potential competition for maternal resources between the toddler that she's carrying, the fetus she's carrying, and perhaps other children that she might have at home. Raising a child is hard work, with costs increasing with the number of children that you have. And there are trade-offs between how much you can invest in each child as number of children increases. In the US, with the cost of tertiary education, you're very, very aware that if you have 4 children, you can afford to invest less in each of them than if you have a single child. So an evolutionary trade-off exists between the number of offspring that a woman produces and maternal effort, investment in each of those offspring. If that were not the case, mothers would have evolved to produce an infinite number of cost-free offspring. Some codfish that produce millions of embryos at a time are getting close to that, but that's clearly not the case in human reproduction. And to understand the trade-offs, some benefits to a child have a cost to its mother's residual fitness, which can be measured in her ability to care for existing offspring or to produce future offspring. And these benefits to the child weighted against the cost to other offspring of the mother are going to be weighted differently in evolution by genes that are expressed in the child or the fetus and genes that are expressed in the mother. So here's a very simple model. I'm having the horizontal axis is looking at the change in fitness to a child or a fetus, and the vertical axis here is looking at the fitness costs to siblings of that child. They could be costs that are mediated through costs on the mother during a pregnancy. And so this simple model is talking about different situations. There are possible changes during pregnancy that are a benefit to the child and benefit to its siblings. Those are unproblematic. Natural selection will favour them. There are events that are potentially costly to child and costly to its sibs. Genetic changes with those effects. Natural selection will tend to eliminate those costly behaviours. But then the interesting situation occurs where there are trade-offs. There are events that could be costly to the child and a benefit to its sibs, or the reverse, benefit to the child, but with a cost to its siblings. An example of that would be a longer interbirth interval is enhancing the child's fitness. So after its birth, it's having longer uninterrupted care from its mother, but it's reducing potentially the number of future offspring that the mother can produce. So looking here, how does natural selection act on genes expressed in mothers? Well, sorry, I jumped ahead. So here we have the unproblematic axis of progress going towards benefits to all individuals in the system. And there is an orthogonal axis of trade-off or potential conflict between different individuals within the family. So genes expressed in mothers evolve to maximise the number of surviving offspring produced by the mother. That's how we think of maternal fitness. So in terms of this simple model, natural selection will favour some genetic change if it enhances the sum of the cost to the child and the costs and benefits to its siblings. So when that sum is positive, maternal fitness is increased, natural selection favours those sort of changes. If that sum is negative, the mother's fitness is decreased, and natural selection tends to eliminate those sort of genetic changes. However, genes expressed in a child evolve to value that child more highly than its siblings, because the gene is definitely present in the child, and it's only got, if we're talking full siblings, a 50% chance of being present in those siblings. So genes expressed in a child will tend to value their own fitness at twice the rate of a cost of the fitness of siblings. This is the expression of sibling rivalry with which we're all familiar. So in this case, natural selection favours if twice the benefit to the child plus the cost or benefit to the siblings is greater than 0. And so I've just indicated here the situations in what is called the child's inclusive fitness is increased, and situations in which the child's fitness is decreased. And those of you who have been observant see that this is not quite the same division of the space that we looked at for natural selection acting on genes expressed in mothers. So there is for the child, for the mother. So this divides the space into regions where there is common interest between mother and child. Either both the mother and child's genes benefit from some change or they both suffer. But then it's also defining areas where there is conflict, where a particular change, for example, during pregnancy, could benefit the child's fitness at the cost of its mother's fitness, or the reverse. So this zone of conflict is where the cost-benefit ratio is sitting, we're talking now full sib families, in the 2-to-1 sort of ratio of cost to benefits to child and to the mother's other reproductive opportunities. So despite broad areas of agreement, there's a lot of selection for cooperation during pregnancy. Most of the evolutionary action will take place in the zones of conflict, because this is where a change of gene expression in the fetus will be countered by a change in gene expression in the mother. So this is where we expect a lot of the evolutionary changes during pregnancy to take place. And to illustrate the costs of these conflicts, I'm going to talk about 3 situations. There are many. One of them is just talking about problems of pregnancy loss. I'm going to talk briefly about menstruation and postpartum hemorrhage, how maternal fetal conflict enters in there, and then I'll finish off talking about the disease of preeclampsia. Now, it was mentioned that I started off as being a botanist. So here is a Banksia, probably familiar to most of you. And I like this picture. So up the top here, we have an inflorescence. So this is a cluster of about 1,000 flowers. And at the base of each of these flowers, there is what a botanist calls an ovary, with a potential embryo developing at the base of that ovary. And when a honeyeater or a honey possum comes along and it spreads pollen over this inflorescence, most of those flowers are going to be pollinated, and most of the eggs at the base of those ovaries are going to be fertilized. So there are potentially thousands of offspring, thousands of embryos in this inflorescence. However, only a very small proportion of them continue development to form fully formed seeds. So this is the big bad Banksia man you're looking at there. Each of those is technically it's a fruit with a well-developed embryo with a seed within that fruit. So here in Banksias, you're looking at about 95% or more of all fertilized offspring embryos are being aborted, and only a very small proportion of them are being carried forward. In fact, that inflorescence couldn't fit all of the potential embryos that are there. Now, botanists have a number of theories what's going on here, what accounts for this very, very high rate of early embryo loss, but a general thought is that because the production of each embryo is very, very cheap relative to the cost of provisioning a seed, that there's been selection for being selective in which of those embryos are provisioned. And I'm going to argue that essentially we expect the same thing to be occurring during human reproduction. The cost of an early embryo is very, very small, whereas the cost of raising a child to independence is very, very large, and so you might expect early on in development for there to be selective processes that are choosing good quality embryos. So here is some classic data from Alan Wilcox. This is looking at women attempting to conceive, and they were using very highly sensitive assays to look for human chorionic gonadotropin in the woman's serum, which was a sign that implantation had occurred. In this study, there were a total of 707 cycles, and of those, they had evidence of implantation in almost 200 of those cycles. Something that we don't know a lot about is what's happening in that attrition from 707 cycles down to 200 implantations. Are those failures of ovulation? Are those failures of fertilization? Or is there a large set of embryos there that have been conceived but aren't even getting to the stage of implantation? Of those 198 chemical pregnancies, only 155 of them resulted in clinical pregnancies. So that was 43 embryos were lost. In the normal course of events, they wouldn't even have been recognised that a pregnancy had occurred. This would just be a delayed menstrual period or a particularly heavy menstruation, which was actually flushing out an implanted embryo. And then of the 155 clinical pregnancies, 136 of them came to term, and the others were miscarriages. And this is a typical figure. About 10 to 15% of clinically recognised pregnancies do not come to full term. So becoming pregnant now, rather than in a month's time, has minimal cost for a mother's fitness. Whereas raising a child of low expected fitness has a major opportunity cost to the mother. And therefore, just as in the Banksia, we expect that mothers should evolve to be risk averse as to which embryos initiate and establish a pregnancy. So some of the early attrition in pregnancy losses is, I think, an adaptive evolved quality control mechanism. It pays for mothers to be selective. If there's any hints that this is not a particularly good embryo, it's perhaps better to try again next month. This is from Macbeth. If it were done, when 'tis done, then 'twere better that it were done quickly. The earlier you eliminate the embryo, the less the cost from doing so. I think that clinical miscarriages, which are occurring much later, are mostly comprised of embryos in which there's a problem that evaded this initial screen on quality. The difference between being chosen and eliminated, of course, though it has relatively little effects on maternal fitness, has bigger effects on the fitness of that embryo, and so we expect embryos to have evolved adaptations to perform well at whatever tests that mothers set for them. It's sort of like an embryo beauty contest at that stage. Interestingly, in IVF clinics, these beauty contests are going on. People peer into Petri dishes or whatever and select the good-looking embryos as the ones to implant. As an evolutionary biologist, what will be the long-term outcome of this? Will we have like peacock's feathers in embryos that are displaying their beauty to the reproductive biologist? So far I've been talking about conventional and adaptive interpretations of pregnancy loss. There is a problem, however. This is a subtle one. A non-inherited maternal haplotype, that's just jargon for a bit of chromosome, gains no benefits from an embryo's survival and reproduction. So a mother has got two sets of genes, and only one of them at random is transmitted to each offspring. The one that isn't transmitted gains no benefit from that particular embryo. Worse than that, because that embryo is consuming maternal resources and time, non-inherited maternal genes would benefit from an embryo's early demise if it was replaced by another that might inherit a copy of those genes. So this is an argument that's suggesting in every pregnancy, half of the maternal genome might be actively conspiring against each particular embryo. Here is an illustration of this. So here, this is representing a maternal genotype, the two versions of a chromosome present in a mother, a yellow one and a light blue one. Here is a different mother, which has a different pair of chromosomes at this genetic locus. Now, under normal events, we expect each of the chromosomes in the mother to have a 50% chance of being transmitted to each particular embryo. It's just like flipping a coin. And so here I'm imagining a very large family of 16 offspring. Half of them on average are going to get the blue chromosome, half are going to get the yellow chromosome. And so we're getting, in the offspring, 8 of each. I'm leaving out paternal genetic contribution here for simplicity. Now I'd like you to consider a different situation. And in this case, we have a mother who has 2 chromosomes at the locus, but the red one makes it hard to establish or implant for embryos that haven't inherited a copy of the red chromosome. So in this case, embryos that aren't inheriting the red chromosome have a high rate of miscarriage, and then they're being replaced by other embryos, some of which are carrying the red chromosome. And so I'm representing pregnancy losses here by the little blue squares. They're the ones that are being knocked off by the non-inherited chromosome that's sitting in the mother. And in this case, we end up with 10 offspring carrying the red chromosome, 2 carrying the blue chromosome, and 8 early pregnancy losses. Now, in this example, I want to point out a number of things. First of all, the cause of pregnancy loss of the blue embryos on the right-hand side is the gene that they didn't inherit. And it's not an intrinsic problem with blue embryos, because on the left-hand side, that side, blue embryos are doing really quite well. Also notice that the mother on the left-hand side here is having 16 offspring, whereas the mother on the right-hand side is having 12 offspring. So there's a substantial cost to maternal fitness here, and yet the red chromosome is doing really, really well. It's present in 10 offspring rather than 8 on the other side. And so this is a simple model suggesting that chromosomes can get to high frequency in the population, even though they're increasing the rate of pregnancy loss, as long as the embryos being lost don't have a copy of that chromosome. This is a strong selective force. So I think effects of non-inherited maternal haplotypes, the genes that a fetus doesn't get, should be considered in common disorders of pregnancy, and especially in cases of early embryo losses. You might ask the question then, how is pregnancy possible, given the argument I've just given? And I think there are 2 factors here. First of these is that this would only work if a chromosome could discriminate among embryos depending on whether they have a copy or not, and for most of the genome is uninformed of that sort of information, so most of the genome is just maximising the number of surviving offspring. Most of the genome in the mother is just maximising maternal fitness and taking a chance on the flip of the coin. The second is that most maternal genes would segregate independently of the red chromosome in this case, and so they're selected to suppress these sort of effects. There are parts of the genome that are involved in self-recognition, and for the specialist here, I'm thinking of the MHC complex and the killer cell inhibitory receptor haplotypes. They possess properties of self-recognition, and so that's particularly where I'd be looking for these sort of effects of selection for early pregnancy loss. Thinking about it, an embryo is in fact non-self to a non-inherited maternal haplotype. It is a foreign invader with respect to that particular chromosome. There's also a danger of blaming the victim. It's not genes that are present in the embryos being lost in this case that are the problem. It's the genes that are not present in those embryos. I now want to switch gears and talk about a second example. This is something that we take for granted as humans, and particularly women, are well aware of. And these are problems associated with menstruation and also, I think, related problems of postpartum hemorrhage at delivery. Menstruation is exceedingly rare among mammals. Menstruation, as occurs in humans, is in our ape relatives and in monkey relatives, and it's essentially unknown among other mammals. There are a few phenomena that have been called menstruation, but essentially most mammals do not menstruate. So this is from Corner, a famous endocrinologist back in the 1940s. Menstruation, he wrote, is a paradox and a puzzle. a normal function that displays itself by the destruction of tissues, a phenomenon seemingly useless and even retrogressive that exists only in higher animals, he means us, and unexplained turmoil in the otherwise serenely coordinated process of uterine function. Now, when you read textbooks, they say, well, menstruation is a consequence of the particular form of human placentation, which is an invasive placentation into the endometrium, and this is a good thing because humans do things better than anything else. That's human chauvinism, and usually they say it's something to do with our large brains or something like that. As I've said, menstruation is restricted to the anthropoid primates, which have these so-called hemicorial invasive placentas. The problem for those arguments that it's adaptive is that a dolphin can produce a large-brained fetus despite possessing a non-invasive placenta without the complications of menstruation and with very little risk of postpartum hemorrhage at the time of delivery. So this is a diagram of a historical embryo described by Peters in Vienna in 1897. So this was a young woman had committed suicide, I think it was a month after a previous period. A pathologist looked in the uterus and found this very early implanting embryo. This was the first evidence that human embryos actually, in a sense, burrowed into the lining of the uterus. And the embryo here is making contact with maternal blood vessels, and as the placenta develops, those blood vessels are opened up and they come to flow through internal cavities of the embryo. Now, I want to suggest that interstitial implantation may initially have been an embryonic adaptation to avoid being one of those pregnancies lost. If it could implant itself in the lining of the endometrium, it couldn't be flushed out by maternal selective mechanisms. Because embryos implant within the endometrium, mothers could not reliably eliminate embryos without shedding the lining of the uterus. And so here we're having this tug of war between maternal and fetal adaptations. So increased depth of shedding at the endometrium was the maternal response to increased depth of implantation. And so this is arguing that menstruation initially evolved as a mechanism of eliminating unwanted embryos that had implanted themselves in the lining of the uterus. Now, as the placenta develops in one of those pregnancies that continue, the trophoblastic cells of the placenta penetrate into the maternal vessels bringing blood to the uterus, the so-called spiral arteries. And during early implantation, the diameter of those arteries is expanded. They're becoming low resistance channels, and also the vascular smooth muscle that causes arteriolar constriction is eliminated. So the blood coming into the placenta from the mother is coming through low resistance vessels that are unable to constrict. Blood from the spiral arteries flows through the intervillous space of the placenta when nutrient exchange takes place, and then it returns to the maternal circulation by uterine veins. This creates an interesting situation where maternal physiology during pregnancy is coming under joint maternal and fetal control because the placenta can release hormones into the maternal bloodstream that are having effects on maternal physiology at remote sites. And we also know that the placenta is releasing fetal cells into the maternal circulation that can potentially be having effects throughout the mother's body. So hemicorial placentation allows maternal physiology to come under partial control of placental hormones coming from the conceptus. Now, a consequence of this form of placentation is that at the time of delivery, the placenta, which is broken into maternal blood vessels, comes away, and you have these large bore vessels that are unable to constrict, and you've got a great risk of hemorrhage at the time of parturition. So at parturition, loss of blood must be staunched by contraction of the myometrial muscle rather than by the absent vascular smooth muscle. And in some cases, this process fails and you have all the risks of postpartum hemorrhage, which is a distinctive feature of human pregnancy. So I'm suggesting that the curses of menstruation and postpartum hemorrhage can be seen as collateral damage of conflict, evolutionary conflict between maternal and fetal genes during pregnancy. I now want to change and finish up talking a little bit about preeclampsia. For the obstetricians in the audience, who's heard of preeclampsia as being the disease of theories? Some of you, yeah. So here is from 2017. Preeclampsia is a disease of theories. There is much that we know, much that we don't know, and even more that we don't know. Okay, so how long has preeclampsia been called the disease of theories? This is the earliest citation I found. So this is from Zweifel in 1895 in German. Preeclampsia is the Krankheit der Theorien, the disease of theories. I actually prefer Tummelplatz der Theorien there, which is sort of the playground or romping ground of theories. So if something has been a disease of theories now for well over 100 years, I suggest that it's a hint that we're thinking about the disease in the wrong way. So what is preeclampsia? It's associated with endothelial damage of the maternal vasculature, which results in vasoconstriction and increase in maternal blood pressure during pregnancy. Multiple maternal organs may fail from oxygen starvation because of this vasoconstriction. So it's a major source of maternal morbidity. Maternal disease is caused by factors that are released into the maternal circulation from the placenta, and the only effective cure for preeclampsia is delivery of the placenta, which cuts off supply of those factors. And for clinicians, here is a classic case of maternal-fetal conflict in preterm preeclampsia. The longer the baby stays in, the better for the baby's health, but the greater the risks for maternal health. So this is a diagram of the maternal circulation during pregnancy. So here I have my poor representation of the maternal heart, which is pumping out blood to the maternal tissues, and a fraction of that blood is routed through the intervillous space of the placenta, and that is providing nutrients to the offspring. So what's going to determine how much of every maternal heartbeat goes to the maternal tissues versus goes to meet fetal demands? It's going to be determined by the relative resistance in the uteroplacental circulation here. So if you reduce resistance here, relatively more blood's going to go to the placenta. If you increase peripheral resistance in the mother, that's going to channel more blood to the placenta. So fetal genes will favour greater blood flow through the placenta than will maternal genes. And so this suggests that the fetal share of mother's systemic blood supply to a first approximation is given by this ratio, the ratio of the non-placental resistance in the mother's peripheral tissues to the sum of the uteroplacental and placental resistance. So there are two ways that the fetus can increase its share of maternal blood supply. One is to reduce the resistance of those vessels that are bringing blood to the placenta. So that's the modification of the spiral arteries we see occurring early in pregnancy. And the second way is to increase resistance in the maternal systemic circulation. If that resistance increases, a greater share of each maternal heartbeat will flow through the low resistance vessels of the placenta. So this leads to a prediction that in pregnancy, placental factors of the fetus will decrease resistance in the blood supply to the placenta, whereas maternal factors will act to increase that. And in the non-placental resistance, placental factors released by the placenta will tend to increase will cause vasoconstriction, increasing resistance, whereas maternal factors will tend to decrease that resistance. And I think that's expressed in the decrease in vascular tone that you see in maternal vessels during pregnancy. So if preeclampsia is a fetal adaptation, it will have multiple functional parts. And a search for the part that is broken will fail. And I think this is the big problem in the last 100 years of preeclampsia research. Everybody has been looking for what is the factor that is going wrong, that is causing the disease, because that's the normal model of disease. Whereas from the factors that are being released by the placenta, I'm saying these are a complex adaptation of the fetus. It's going to be an adaptation with multiple parts, and so we're sort of looking in the wrong place in understanding what is happening in this disease. So more generally, maternal-fetal relations lack important feedback controls that we see in other physiological processes because signals from mother to fetus lack evolutionary credibility because of the conflict of interest in an evolutionary sense between the two generations. So grant proposals in obstetrics and gynecology are full of maternal-fetal dialogue and the intimate exchange of messages between mother and fetus. And I think there's actually relatively little evidence for that detailed sort of communication. Think of an ectopic pregnancy. An embryo will develop in completely inappropriate places within the maternal body. So I think some of the problems that we see in pregnancy are a lack of homeostatic feedback controls. The physiology of pregnancy is inherently unstable because of failures of communication. So maternal-fetal dialogue in this model resembles more a political debate. And political debate, as we know, is rarely smooth and efficient. Physiological processes are less efficient in pregnancy than other metabolic states because of unresolved evolutionary conflicts, and maternal pathology may be an expression of fetal physiology and vice versa. And with that, I'd be happy if we have time to answer any questions you might have. Thank you.
Well, thank you, David, for that really remarkable exposition of the inside of your brain, which I think we all would agree is going to give us new ways of considering what to do about these really intractable conditions. Questions for David? Can I start by asking you, and I think you made a very good case for why it's important that mothers should be choosy and exert quality control.
Yeah.
Is it important that fathers should evolve genes and processes to encourage the mother to be choosy, or should the father be convincing the female to accept anything?
So let's not talk about fathers, but talk about paternal genes within the embryo. So my work on, so one thing that I simplified here is my work on imprinting is actually looking at conflicts between genes of maternal and paternal origin that are expressed in the fetus. And there's good evidence there that paternal genes, for example, are favouring relatively greater development at the placenta, which is the feeding tissue, and they're being restrained by genes that the fetus inherits from the mother. So there is selection on embryos. If an embryo is of really low quality, it could be selected to fall on its sword for the benefit of a sibling with higher quality. And for maternally expressed genes, that turnover point is when the 2-to-1 ratio. If the other embryo has twice the quality of this one, the embryo should favour its own demise. In situations where you have partner change, the paternally derived genes in the embryo are less likely to be present in the next embryo, and so they are particularly engaged in suppressing those sort of mechanisms. So I think there's an actual conflict within the developing embryo between the genes it gets from its mother and father on pregnancy losses. Yeah.
Thank you. Wonderful talk. Thank you. Wonderful talk. First of all, I just want to say one thing briefly about embryonic and fetal loss,
Yeah.
clinicians in this room and elsewhere, of course, fight to save these embryos, but really from what you're saying is we shouldn't actually be saving them.
So I think a lot of embryo loss is part of this selective mechanism, but because the cost, Randy Nesse has called this the smoke alarm principle. So we know the risk of making a mistake, of not evacuating the house when it's on fire, is really large. And so smoke alarms tend to go off in lots of situations where an embryo, where there isn't a fire. And the same thing, I think, is happening in early embryos. So quite a lot of the early embryos that are being lost are probably quite viable embryos. It's just that it's a very risk-averse system. But in assisted reproductive technologies, when you are suppressing the methods of pregnancy loss, I think you're going to increase the frequency of births with embryos that have problems because that quality control device is absent. Now, some pregnancy losses, as I've suggested, are coming about by these insidious haplotypes and they're not adaptive, that are doing in embryos without that copy. And then every system also has its pathologies and its failures. And so some women with recurrent pregnancy losses, there's some underlying maternal pathology, even though the embryos might be good. So it's a complicated issue. And I was just talking to Michael Davies from here in Adelaide. There is very good evidence that the rate of fetal abnormalities is dramatically increased in IVF pregnancies. There are many factors accounting there, but part of it may be that embryos that would have been eliminated are getting through screens.
But also Michael would contend that some of the drugs that are used may be causal.
Yeah.
Can I also ask you, well, first of all say, I lost count of the number of times you said placenta. Yay! But your conclusions at the end about these maternal adaptations and fetal signalling, all these complications, not just preeclampsia, we should be thinking then, and I have argued this, they're not one thing.
Yep.
The gestation you're born at just happens to be the gestation you're born at, doesn't say anything about the pathway to that. Yeah. Similarly, with preeclampsia, we should be thinking of it as a syndrome with multiple pathways, that some of which interact, some of which don't, and in some women and some fetuses occur differently.
Yeah.
So, of course, that means when we're trying to look for one causal factor, our favourite gene or whatever it is, we're not going to succeed.
Yeah.
So what do you think we should be doing then? What can we do if we know there are these multiple pathways? Yeah. Can we really do something about these problems?
So what I'm trying to, I'm becoming a tedious fellow talking. I've been talking about preeclampsia now for almost 30 years. Evolutionary theory is not giving a solution to the problem. That's clinical research is going to get there. But I think some of the problems is clinical researchers have theoretical positions they're taking that they're dealing with a cooperative process. They're not even aware that they're making those sort of assumptions. And so I'm trying to relax those assumptions. So in preeclampsia, I would want to understand the nature of the fetal adaptation. What are the coordinated parts? In particular, what are the events that trigger activation of that pathway? I would like to, as you're suggesting, separate preeclampsia into a lot of different conditions. In some, the underlying problem is in early implantation and a poor blood supply. In others, in Ecuador, the suggestion it's calcium deficiency. And so I think all sorts of different nutritional disadvantages can trigger a fetal response to increase the supply of blood to the placenta. So this model is suggesting that preeclampsia will have a whole variety of different environmental causes.
Could you say something about pre-fertilization competition? Because I think that's been one of the areas that you've been well known in terms of sperm competition.
Yeah.
But is there also competition among eggs to get there? And is this an important physiological evolutionary factor?
So the example I gave of that red haplotype that is causing the abortion of some embryos, and you can think of it of the abortion of some eggs for the advantage of others, that in the formal evolutionary model, that's very, very similar to a model of meiotic drive, where you have some sperm in the ejaculate killing other sperm in the ejaculate to get to fertilise eggs. And so now under my theoretical hat, these are all just forms of distortion of the fairness of genetics, of one gene getting an advantage of the other in what you would call an unfair way. And so those events can also occur during, potentially during oogenesis. But an important thing to say about oocytes are diploid, so they're the maternal genotype. They've got both alleles. First meiosis is completed just before ovulation, and second meiosis, which is reducing to the haploid stage, is actually occurring after the sperm has entered the egg. So the possibilities of competition, so there isn't competition among oocytes. Where you see this competition is between chromosomes of whether they're being expelled in the first or the second polar body. And Hamid Malik, for example, has found evidence that there is competition among centromeres that is affecting their probability of going to, and in fact, what's his name? Anyway, there's beautiful work being done on that in Adelaide, and here you are, Keith. Keith is doing work on that and is finding your executive dean, right, good. Anyway, so talk to Keith later about that, good.
Yeah, my problem is that I've listened to your lecture and I was fascinated. The problem is that I always see reports in the paper about women who have lost their fetus or whatever, that they've had a miscarriage or whatever, and yet there seems to be still within that thing that it's their fault, that we don't do enough for telling them that these things are perhaps normal. And that's all. That's what I want to say.
Yeah. So if we so the pregnancy losses that people are aware of are those clinical miscarriages, and the general result is about 10 to 15% of pregnancies are lost. And in the majority of those cases, there's not an underlying problem in the mother, and they're likely to conceive again. So fertility clinics only start worrying when a woman has had particularly 3 losses in a row. It's, if in a large population of women, if 10% are being lost, some women will just have 2 losses in a row just by bad luck. 1 in 100 women would have it. It's when you're starting to have a very large number of losses that there's a suggestion that there's some sort of underlying problem, and it's not a problem with the woman, it's just it's maybe some environmental exposure, or it's just some genetic variation, like some of us are more prone to diabetes than others. That would be an example, yeah. So there was one up there.
Thanks. So if an invasive placenta is the result of the mother-offspring conflict, why do we only see them in anthropoid apes? Why don't we see them more widely across animals?
In fact, many mammals do have different forms of invasive placentas. In fact, the placenta is the most variable organ among mammals, and I think that's because of these conflicts, because this is you don't settle on an efficient outcome, so the placental form is constantly changing. And it would be a long story to talk about. So mice have an invasive placentas, but they don't eliminate embryos by shedding the lining of the uterus. partly because they're producing a litter of many offspring at one time, they have processes where some embryos within the litter are resorbed and consumed by maternal tissues. So, you know, it's sort of hard to generalise from one mammal to another. I teach a course called Vertebrate Viviparity at Harvard, and I have one of my favourite lectures is on sharks. And 60% of sharks give live birth. Some of them produce placentas, but one of my favourite forms of nutrition in sharks is the group to which the great white shark belongs. And the mother just keeps on ovulating during pregnancy, and the fetus sits there and has eggs breakfast. And over a 9-month pregnancy consumes about 1,000 eggs. In the grey nurse shark, the embryos don't stop at eating eggs. They practice what is called adelphophagy. If you know your Greek, this is consuming your brother, or intrauterine embryonic cannibalism. And so you start off with about 4 embryos in each uterus, and only one of them survives after consuming his brothers and then starting to consume eggs. So I'm absolutely fascinated by pregnancy. As an evolutionary biologist, I just think it's the most interesting relation out there, because it's also one of the most intimate interactions and prolonged interactions there is between 2 genetic individuals. And like any complex interaction, it's going to have elements of cooperation and conflict. You know, is a marriage cooperative or conflictual? Well, it depends. Yeah.
That might be a good place to finish.
Okay.
David, we would like to thank you so much for making the effort to come to Australia and to give this fascinating lecture. And I think everyone will agree the journey you've given us from Banksia to women to menstruation and mice and sharks is so thought-provoking and so exciting. So sincere thank you, and all the best for these fabulous theories as they evolve in the future. Thank you.